So now we got our monomers, okay? And remember the patterns. Typically, you go from big molecule in the mouth to smaller molecules and then to individual monomers at the brush border, right? That's usually the last step is at the brush border. Okay, so now we've got our products of digestion. We have our simple sugars, our amino acids, our fatty acids, and our monoglycerides. All right, so now we got to talk about translocation. What I mean by that is moving it from the lumen, the material, from the lumen into the blood where it can be useful to the body. All right, you've probably talked about some of this in anatomy, but I like to talk about it in terms of surface area expansion. Absorption is happening primarily through diffusion and co-transport. Okay, we're going to look at a number of transporters here in a minute. That means that it's the cell, it's the membrane, it's the surface of the cell that's doing the busy work of absorption as well as digestion. So if the surface is doing the work, we need a lot of surface, right? So we have surface expansion. The first layer are the folds of Kirkring, right? When you look macroscopically at the small intestine, it literally has folds you can see, right? It's like a wrinkled sheet. Now, if you zoom in and look at an individual fold, the individual fold has a hill, right? That's the villi, okay? There are more folds and villi early in the intestine and fewer later. It's because most absorption occurs early and less occurs later. So we have more surface membrane. We have more membrane in the front half of the intestine than the back half. Then if we zoom in and look at the villi, we see that the villi have microvilli on top. That's the brush border. Now this is all membrane, okay? So when we're going to look at our pumps and transporters in a minute, this is the membrane where it's living on. So there's a lot of it. There's a lot of membrane, there's a lot of cells because of the villi, and there's a lot of villi because of the folds. You see the pattern. So all told, it's 1,000-fold expansion of surface area, about the size of a tennis court, or surface membrane expansion. Reminder about the villi. Okay, two pictures. This is cross-section. This is from the side, right? So we have our arteries going up, veins coming down. Remember, oxygen can bypass, right, because of this parallel arrangement. But what I want to point out to you today is right through the center is a big fat lymphatic called a lacteal, all right? And it has the same root, lactus, is milk, right? Like lactose is milk sugar. And the reason it has lactus in it is because what comes out of here is typically white, like milk. Because what comes out of the lacteal is lymph with a whole bunch of dissolved fats in it. All right, specifically fats that have been packaged into chylomicrons, which a chylomicron is kind of like an M&M. You know, you got fatty stuff in the middle, and then you have this sort of outside coating that's more friendly to the environment. Well, a chylomicron is the same way. The surface is polar, so it can dissolve in water. The center is non-polar, so that's where all the fats are. We're going to pick up the chylomicron story later. But that's what's coming in through the lacteals. Okay, so we can see there's sort of a central lymphatic.